So I wanted to really give this first talk on the pilot study because you're right at the very beginning of the path for developing a biomarker signature when you're designing your pilot study. And so that's the focus today. There are some things that you can do early on that will make the entire path more efficient and that will help accelerate you through the path to clinical validation. Each step of the process is challenging in its own right, but I really believe that the pilot study is the most challenging because usually you know the least about your patients prior to the pilot study, and so when you're putting together your study design, at this point in the path, you basically have the least amount of information to go on, and that can be particularly challenging. But I really believe that if you focus on doing the pilot study and the confirmation in a manner that produces a robust signature, it will actually help accelerate you through establishing the analytical performance and ultimately the clinical validation and demonstrating the clinical utility. So let's get started. Good scientific method. I know all of you are familiar with this because you wouldn't be joining us today if you weren't. Everyone learns this when they're doing their training, and these are basic principles that people are familiar with, but it's a very good exercise to go back and revisit these and really have a checklist to make sure that you've covered all aspects, to make sure that your plan is going to hold up under the scrutiny and the scrutiny of your peers, and also that you can justify your expectations versus what you actually observe at the end of the study. So first of all, clearly defined goals and milestones, which I mentioned earlier. Especially when you're looking at whole microbiome studies, it's a lot of data. It's analogous to a whole genome study or a whole genome expression study. You're going to be faced with a tremendous amount of information. And so you need to be clear at the outset if you're going to be looking at the fish or the birds or some combination, or are you comparing attributes of fish and birds, which is why I like to use this Escher image when I talk about clearly defining your goals and staying focused on the question that you're particularly interested in. Secondly, good scientific method, you want statistically sound study design. So it's imperative that you have some statistical support and people who are knowledgeable about dealing with this amount of data when you're setting up your study design. Now, one of the challenges with a pilot study is that you generally don't have much data to go on. So making an estimate with respect to how to statistically power your study is really a back-of-the-envelope rough calculation. And I'll talk a little bit later about the things to consider when you're setting up your pilot study that can help you build in some contingencies to make sure that at the end of the pilot, you've got an adequately powered study. So early on, you project the correct number, but you don't actually know until you have data, and we'll talk about that. And that leads into the next point, meaningful number of each sample type, making sure that your cohorts are well annotated and that you understand how they've been characterized. You want to use an accepted analytical method that's scientifically justifiable. I think that goes without saying. You want to use a robust platform, a platform that is well characterized so that you know what the sources of variability may be that are associated with the analysis method. And then part of your team needs to be someone who knows informatics, the bioinformatics, the analysis methods, and data management that will help you not only have well-annotated metadata, but that it will be accessible. So that's the overview of general scientific method, and now I'm going to address key features of a pilot study. So these are some of the key considerations for a pilot study. You want sound quality control practices following best practices, established best practices. There are lots of protocols out there. There are established best practices for quality management systems and handling and tracking the samples. You want to have, basically what you want to know is the life cycle of every one of your samples through all of the processing steps. Because if there's a deviation from the protocol, and that happens for some samples, if there's a deviation, you want that annotated, because later on when you're doing your data interpretation, that can actually impact the way you interpret your data. So for instance, if you see an outlier in a cluster or a group, you really want to know, is there something special about that patient, or was there something about the way that sample was handled, or perhaps it was a low biomass sample, or perhaps it didn't PCR amplify well. You want to be able to know that, because it will impact your interpretation of the data. And if you need to filter your data and remove samples because of some laboratory artifact or sample artifact, that should be tracked. Secondly, quality management documentation. I use this image of Leonardo da Vinci's notebook. If Leonardo wouldn't have written things down, we wouldn't have any idea that any of these wonderful ideas existed. And so I'm really a stickler for tracking and making sure that you know the location of every sample at all times. Sample mix-up can kill a study, and that's why I also have on this slide a barcode. It's really important to have a tracking system in place, especially to build on. So you start with a smaller pilot study, but eventually, if you're successful, you're going to be building on that data set, and you want to have a system in place so that you don't have to worry about things like sample mix-up and the life cycle of the sample. You really want to focus on the science. The analytical performance of the assay should be well characterized. You want to understand the repeatability and reproducibility of the method. Repeatability simply means if you ran the same sample in the same lab, how close would the results be? If you took the same sample and split it between labs and ran it in two different places, how close would the results be? That's a source of variability. If those sources of variability are larger than the differences that you expect to see between the two groups that you're comparing, group A and B, for some biological variant that you want to determine for establishing your biomarker panel, if those analytical performance differences are large, you will have a very difficult time extracting a biomarker panel that's robust. And then finally, you want to avoid pooling. I know it's not always possible to do this, but pooling will vary information that could be very important to either understanding the etiology of disease, or if you're doing discovery and you want to really understand all of the taxa that could be involved in, say, response to treatment. Pooling will basically dilute those, and you don't want to overwhelm those low biomass numbers that may be important for your understanding of the biology underlying the disease, or creating a biomarker panel that is effective. So this is something that I know sometimes it's just not possible, and there are times when it's appropriate to pool, but not during the biomarker discovery phase of the study.